Imagine for a second that you're tasked with understanding exactly how a massive, towering skyscraper was built from scratch.
Okay.
Like from the ground up.
Oh, wow. Okay, that's a big job.
Yeah, but here's the catch. The only piece of evidence you're given is a single two-dimensional photograph of a wall.
Just a wall.
Just a wall. You have like no blueprints. You don't have a time-lapse video of the cranes moving or the steel girders being placed. You just have that one flat, static picture. And I mean, for a very long time, that incredibly frustrating scenario is essentially how scientists have had to study organ formation in biology.
Yeah, it's painfully accurate. I mean, we've historically relied on taking these extremely thin two-dimensional slices of tissue, right? You put them on a glass slide, and then you just try to infer the dynamics of a three-dimensional living biological structure.
Which seems impossible.
Right, because you might capture a snapshot of a single moment, but you lose the entire spatial architecture. And more importantly, you completely lose the dimension of time.
Exactly. Which brings us to the mission for today's deep dive, because today we are unpacking a truly groundbreaking paper. This was published in the journal Cell in September 2026 by Asami and colleagues.
Truly a paradigm shift, this paper.
It really is. We're looking at a revolutionary new technology that takes a single three-dimensional chunk of tissue and somehow, and we're going to explain how, turns it into a four-dimensional, high-definition movie of organ development.
The level of mechanical detail we can now see is just, it's staggering. I mean, we're moving from educated guesses based on those flat 2D slices to actually watching the exact cell-by-cell choreography of how a mammalian organ builds itself over time.
Okay, let's unpack this technology first, because frankly, it sounds a little like science fiction.
It does, yeah.
The paper introduces a process called 3D Deep. So that stands for 3D DNase-Enhanced Expression Profiling.
Right.
And the core problem it's trying to solve is, well, how do you read RNA deep inside a thick piece of tissue without physically slicing that tissue into tiny pieces?
Right. So to do that, the researchers have to clear out all the biological clutter.
The clutter.
Yeah. So they take a thick block of tissue, and for this specific study, they used newborn mouse skin. And they subject it to this really exhaustive, multi-step chemical process, because the goal is to make the tissue physically transparent to chemical probes.
Okay.
Which means they have to actively remove the genomic DNA, the proteins, and the lipid fats that make up the cell membranes.
Wait, I need to pause you there. If you introduce chemical detergents to, like, strip away all the structural fats and the cell walls and the heavy proteins, why doesn't the entire tissue just disintegrate into biological soup?
That is the big question.
Right. I mean, if you dissolve the structure, why don't the RNA molecules we actually want to study just wash right down the drain?
Well, that is the crucial first step of the 3D process.
Yeah.
Before they wash anything away, they embed the entire block of tissue in this clear polyacrylamide hydrogel.
Polyacrylamide hydrogel. Okay.
Yeah. You could think of it as injecting the tissue with a liquid resin that polymerizes or hardens into a microscopic three-dimensional mesh.
Oh, I see.
And this chemical spider web physically links to the specific molecules we care about, which in this case is the messenger RNA. And it anchors them in their exact 3D coordinates.
Wow.
So when the enzymes and detergents wash away the cell membranes and the genomic DNA, the mRNA remains suspended in this clear matrix. It perfectly maintains its spatial orientation.
That makes total sense. But it brings up another question for me. You mentioned they use an enzyme, DNase, to remove the genomic DNA.
Yes.
Isn't the DNA the master blueprint? Like, why on earth would you intentionally destroy the most important part of the cell?
It's a great question.
Yeah.
So DNA is the master blueprint, but it doesn't actually tell you what the cell is doing at that specific moment.
Okay.
So remember, every cell in your body has the exact same DNA archive. But messenger RNA, or mRNA, those are the active work orders.
The work orders.
Got it. Right. So if a cell is building, say, a hair shaft, it sends out mRNA work orders to produce keratin. By destroying the bulky DNA archive, we free up physical space inside that hydrogel mesh so our chemical probes can penetrate hundreds of microns deep.
Just to read the active stuff.
Exactly. We only read the active real-time work orders.
So it's kind of like pouring clear resin over a massive, incredibly messy library. You anchor the specific books that are currently open on the desks, and then you completely dissolve the bookshelves, the dust, all the closed books.
That's a perfect analogy.
And you're just left with this transparent room where only the active reading material is floating in space.
Yep, exactly.
But how do you actually read those floating mRNA work orders, though? I mean, they must be incredibly small.
They are tiny. which is why the team uses something called padlock probes.
Padlock probes.
Right. These are custom synthesized strands of DNA that are designed to hunt for a very specific sequence of target mRNA. And when a probe finds its exact match floating in the hydrogel, the two ends of the probe wrap around the mRNA strand and physically snap together.
Oh, wow.
Yeah, they form a closed, continuous circle. So it literally acts like a microscopic padlock clamping onto the target.
But I'm assuming a single microscopic padlock is still way too small to see, even under a powerful microscope.
Oh, yeah, much too small.
Okay.
So to make it visible, they trigger a biological amplifier. It's called rolling circle amplification, or RCA.
RCA, okay.
Once that padlock is snapped shut, an enzyme latches onto the closed circle and just starts reading it. And the enzyme just continuously runs around and around the track, generating this long, repeating tail of DNA based on the padlock sequence.
So like a loop.
Exactly. It eventually creates a tightly packed repeating ball of DNA that is hundreds of times larger than the original mRNA target.
So it's essentially a biological printing press. It takes one tiny signal and just cranks out copies until it creates a massive clump.
That's exactly it. And once you have that massive clump, you wash fluorescent tags over the tissue.
Oh, to make it light up.
Right. These tags are designed to bind only to those giant RCA DNA balls. So suddenly, the specific mRNA work order you want to read is glowing neon bright under the microscope, perfectly preserved in its original 3D spatial location.
That is wild. You can map exactly what every cell was doing at the exact moment the tissue was harvested.
Yes, exactly.
Okay, I am tracking with the 3D map part. We have the X, Y, and Z axes. The paper insists on calling this a 4D map, meaning 3 dimensions of space plus the dimension of time.
Right.
If this whole process starts by taking a single frozen chunk of newborn mouse skin, how do we get a movie out of a static photograph? Like, where does the time come from?
So this is where the computational side of the study really takes over. They utilize a concept called spatial pseudo-timing.
Spatial pseudo-timing. Okay, walk me through that.
Well, newborn mouse skin is a highly unique environment. At the exact moment of birth, the skin is packed with hundreds of individual hair follicles. But, and this is the key, they do not develop in unison. They grow completely asynchronously.
Meaning they're all at different stages.
Exactly. In a single square millimeter of tissue, you might have one follicle just starting to form, another that is halfway developed, and a third that is nearly fully mature.
So the researchers have a single physical block of tissue, but inside that block, they have hundreds of biological clocks all ticking at different speeds.
Yes. And because these follicles grow continuously, their physical length strongly correlates with their developmental stage. So the researchers painstakingly measured the exact physical length of hundreds of follicles in that 3D block.
Okay.
Then, using the 3D kit technology we just discussed, they looked at the complete molecular profile, the transcriptome of every single cell in those follicles, and they fed all of that massive molecular data into a mathematical algorithm called principal component analysis, or PCA.
Wait, I've heard of PCA. It's used in data science and finance, right? To find patterns in huge spreadsheets.
Yes, it's a very common tool there.
But how does a math equation tell you the physical age of a biological cell?
Well, PCA is fundamentally an algorithm that looks for variance. When you have a data set with thousands of variables, in this case, thousands of different mRNA levels, PCA identifies the primary axis where the data differs the most.
Okay, so it looks for the biggest changes.
Right. And in a developing organ, the single biggest source of molecular difference between any two cells is their level of maturity.
Ah, because a young cell is sending out completely different work orders than an old cell.
Exactly. The algorithm doesn't know what a hair follicle is. It just looks at the math and naturally arranges the molecular signatures from the most biologically immature state to the most mature state.
That is brilliant.
So by plotting the physical length of the follicles against this calculated molecular age, the researchers could line them up in a perfect sequential timeline. They reconstructed the fourth dimension time entirely from a single spatial snapshot.
Just to make sure I'm wrapping my head around this concept before we move on, think of it like taking a single wide-angle photograph of a really busy school playground.
Okay, I like this.
Right, so in that one split-second snapshot, you capture toddlers in the sandbox, young kids on the swings, and teenagers playing basketball.
Right.
If you physically cut out their individual photos and line them up strictly by their height and physical maturity, you would create a perfect, seamless time-lapse of human growth, all derived from a single static photograph.
That is spot on. You were capturing an entire developmental lifespan in one frozen frame. They built a 4D time machine out of a single piece of tissue.
Which is just an incredible engineering feat. So before we jump into the next part, let's just do a quick recap for you listening, kind of bullet point what we've covered so far.
Sounds good.
So bullet 1, 3DP clears genomic DNA to read RNA and thick 3D tissues using that hydrogel mesh. Bullet 2, rolling circle amplification, the RCA, makes the RNA visible like a biological printing press. And bullet 3, spatial pseudotiming sorts asynchronous structures by age to simulate time, giving us that 4D movie.
Perfect summary.
And it makes me wonder, for you listening right now, if you had access to this 4D time machine today, which organ or system in your own body would you want to freeze and map out? I mean, think about that. Play and watch the choreography of organ assembly unfold.
It's pretty amazing.
So let's look at what the paper found when they actually mapped the hair follicle.
Right. So the hair follicle might seem like a trivial organ compared to, the heart or the brain, but it is an incredibly complex microorgan.
Okay.
It's composed of 3 entirely different cellular lineages that have to coordinate perfectly. First, you have the main building blocks, the ectoderm lineage, specifically the keratinocytes. Yeah. These form the physical walls, the structural sheaths, and the actual hair shaft itself.
The bricks and mortar.
Exactly. Second, you have another ectoderm lineage, the melanocytes. These are specialized cells that migrate into the structure to provide the pigment,
giving the hair its specific color.
Yes. And finally, you have the mesoderm lineage, the fibroblasts. These form the surrounding dermal sheath and a crucial cluster at the base called the dermal papilla.
And what do they do?
The fibroblasts provide the structural scaffolding and, more importantly, they broadcast the chemical signals that tell the other cells what to do and when to do it.
Okay, so we have the building blocks, the paint, and the structural engineers all trying to talk to each other.
That's a great way to put it.
So the researchers map this development across a pseudo-time scale, running from 0 to 1. They define stage 1 from pseudo-time 0 to 0.3 as patterning and stratification. Now, logically, if I'm building a house, I pour the concrete foundation, I put up the wooden framing, and then the specialized workers like plumbers and electricians move into their designated rooms.
Yeah.
Does the 4D map show the cells building the physical structure first?
What's fascinating here is that the 4D mapping revealed the exact opposite.
Really?
Yeah. During stage 1, the physical structure of the follicle is incredibly primitive. It looks like a simple downward nub of tissue.
It's just a nub.
Yeah. There is no bulb, no complex layers, no hair shaft. Yet, the molecular map shows that the stem cell compartment, the specialized cells that will eventually maintain the adult follicle, is already stratifying and organizing itself along the vertical axis long before the complex structure is built.
Wait, really? So the roles are assigned and the cells are spatially locked into their designated zones before the factory even exists?
Exactly. The cells are reading their environment and positioning themselves based on invisible chemical gradients. They establish their spatial coordinates first, and that allows them to execute the heavy construction phase, which is stage 2.
Okay, stage 2.
This runs from pseudo-time 0.3 to 0.7, and the researchers call it diversification and bulb formation.
So this is where that simple nub of tissue just explodes into complexity.
You got it. The simple early structure rapidly diversifies into highly specialized concentric layers. We see the emergence of the inner root sheath, which acts as a rigid physical mold for the hair.
Okay, the mold.
We see the hair shaft itself begin to form within that mold. We see the companion layer forming a boundary. And critically, we see a fascinating mechanical movement involving the fibroblasts we mentioned earlier. A cluster of these fibroblasts, called the dermal condensate, physically invaginates.
Invaginates, meaning it folds inward on itself.
Right. It pushes up into the bottom of the developing follicle, creating a pocket. This cluster becomes the dermal papilla, which is essentially the command center for hair growth. If this physical invagination doesn't happen perfectly, the entire organ fails.
Wow. So stage 2 is the heavy construction phase. You get the mold, the shaft, and the command center.
Yep.
But then we hit stage 3, running from pseudo-time 0.7 to 1.0. This is growth and canal formation. And here's where it gets really interesting to me.
Yeah, this part is wild.
Because I always pictured a growing hair just like violently drilling its way upward through the skin like an oil derrick. I mean, how does the hair actually break through?
So the 4D map shows us that biology is much more elegant than brute force drilling. The researchers discovered an incredibly specific choreography to create the exit route, which is called the hair canal.
The hair canal.
It is not a single tunnel being dug from the bottom up. Instead, the data shows 2 completely separate hollow cavities emerging independently of each other.
Independently?
Yeah. One cavity forms high up near the surface of the skin, and a second cavity forms lower down, deep within the follicle structure.
Two completely isolated pockets of empty space. How do they ever connect?
It's the biological equivalent of a coordinated engineering project, driven by the synchronized remodeling of three specific tissue layers: the inner root sheath, the companion layer, and the upper follicle keratinocytes. These cells physically rearrange themselves.
Okay.
This causes the lower cavity to expand upward, while the upper cavity simultaneously descends. Eventually, these two isolated spaces perfectly intersect to form a single continuous canal. This clears a completely frictionless path for the hair shaft to emerge.
It is exactly like how they built the Channel Tunnel between England and France.
Oh, yeah.
Right. You have a massive physical barrier, so you start digging from two completely separate sides, one team in the UK, one team in France. And through precise mathematical coordination, those two massive drills perfectly meet in the middle beneath the ocean to form one continuous passage.
And just like the Channel Tunnel, if the molecular surveying is off by even a fraction of a millimeter, or if the timing is delayed by a few hours, those two biological tunnels will miss each other.
Oh, wow.
The canal fails to form, and the hair becomes permanently trapped beneath the skin.
Let me just pause and reflect on this, because the level of coordination is mind-blowing. Let's do another quick recap for you listening. Bullet 1. Stage 1, the stem cells act like project managers, stratifying and organizing early roles defined before structure.
Right.
Bullet 2. Stage 2 is rapid diversification into complex inner structures and the bulb. And bullet 3. Stage 3, the canal forms by two separate cavities perfectly merging.
Spot on.
It actually makes you wonder, think about a complex project you've managed or a team you've led. Did you clearly define the roles and the structural hierarchy before you started laying the physical foundation, just like the hair follicle stem cells do?
That structural coordination is everything. I mean, we now have the complete blueprint of a perfect, synchronized biological ballet. But the ultimate test of this 3D technology isn't just watching things go right.
Right, it's what happens when things go wrong.
Exactly. It's asking, what happens to this magnificent choreography when a single piece of genetic code is altered?
So to figure out how the system breaks, the researchers applied their 4D mapping to a specific genetic mutation. They used the FOXN1 deficient mouse. Now, in the scientific community, this is commonly known as the nude mouse because it appears completely hairless.
Yep.
And I always assumed a nude mouse just lacked the biological program to grow hair entirely, like it just didn't have the code.
It's a common misconception. For decades, the nude mouse has been a critical model in immunology and dermatology, but the actual mechanics of why it is hairless were deeply misunderstood.
Really? What was the old view?
The old consensus, which was based entirely on those standard 2D tissue slices we talked about, was that the hair follicle actually formed perfectly normally for most of its development.
Oh, I had no idea.
Scientists believed it just suffered a focal structural breakdown at the very end. The hypothesis was that the hair shaft just got structurally weak, folded over on itself, and failed to push through the skin.
But when the Asami team ran the tissue of these mutant mice through the 3D time machine, they saw a completely different reality. The problem wasn't at the end of the assembly line at all, was it?
Not even close. The 4D map revealed that the late-stage structural collapse is merely a symptom of a much earlier, deeper systemic failure. By tracking the molecular pseudotime, scientists discovered that the core genetic program for building the follicle is present, but its execution is fundamentally corrupted from the very beginning.
Okay, but what is the actual mechanism of that failure? Like, what does a corrupted execution look like at the cellular level?
The mapping highlights a profound, lagging molecular progression.
Lagging progression.
Yeah. In a healthy organ, cells proliferate, meaning they physically multiply and grow in number at the exact same time they mature and differentiate molecularly.
Right, so size and maturity match.
Exactly. But in the mutant mouse, these two processes decouple. The cells are physically multiplying and getting larger, but their molecular maturity is severely delayed.
So they are growing in size, but they aren't growing up. They don't know what their specialized jobs are supposed to be.
That's a great way to frame it. And this leads to massive developmental instability. In a normal mouse, every single hair follicle hits its developmental milestones at the exact same pace, like clockwork.
Right.
In the nude mouse, the map showed wild variability. Some follicles were developing fast, some were incredibly slow, and others were just stuck in a loop. There was a complete loss of organ-wide coordination.
So if we connect this to the bigger picture here, it completely changes how we view single gene mutations. Because the mutated gene, in this case, FOXN1, is normally only highly expressed in very specific cells, like the mature matrix or the hair shaft itself.
All right.
But the ripple effect of that one broken gene is staggering. The 3D map proved that up to 11% of the probed genes in the fibroblasts were significantly altered.
And keep in mind, those fibroblasts don't even express the mutated FOXN1 gene themselves.
Wait, they don't?
No, they don't. They are just the structural scaffolding and signaling cells. But because the mutation in the primary cells alters the global signaling environment, the fibroblasts start receiving garbled instructions. The entire communication network of the organ breaks down.
Wow, so the old 2D view thought the failure was like a single violinist's string snapping at the very end of a concert, causing a minor disruption in the finale. But this 4D map reveals that the entire orchestra was playing slightly off-tempo, ignoring the conductor, and playing entirely different pieces of music from the very first movement. By the time they reached the finale, the entire symphony was a chaotic mess, and the structure collapsed.
That is exactly what's happening. And that profound insight is only possible because we can track the exact molecular state of every cell in 3D space across time. We can actually see the biological orchestra falling out of sync long before the music stops playing.
It's honestly terrifying when you realize that an entire systemic collapse can be traced back to a tiny loss of coordination at the very beginning of the process.
It really is.
So let's do our final quick recap. Bullet 1. Nude mice hair loss isn't just a late-stage structural break. Bullet 2. 3D reveals early lagging progression and massive discoordination. And bullet 3. Genetic mutations in one cell type cause massive ripple effects in the global signaling environment. And it actually makes you wonder whether you work in software engineering, in corporate business, or just managing a household. Have you ever seen a tiny, almost invisible timing delay early on cause a catastrophic, unfixable failure at the very end of a project?
It is a universal principle of complex systems, whether they're biological or artificial. Timing, pacing, and spatial coordination are just as important as the physical components themselves.
So bringing this all together, what does this all mean? We didn't just spend this deep dive talking about the technicalities of bald mice. Why does tracking the molecular pseudotime of a hair follicle matter so much to the broader medical and scientific fields?
Because the hair follicle is the ultimate proof of concept. It is a highly accessible mammalian organ that undergoes continuous regeneration. By proving that 3D can successfully create a complete 4D map of the hair follicle's assembly and its failure modes, the researchers have set a new paradigm for understanding any complex organ assembly.
Oh, I see.
If we can map the skin, we can adapt this to map the assembly of the kidney, the development of the lung, or the intricate wiring of the brain.
Which gives us a brand new high-definition lens to understand human congenital anomalies. I mean, think about complex conditions like Charge syndrome, where human organs do manage to form, but they have subtle, highly variable, and often devastating malformations.
Exactly.
Previously, we only had static pictures, so we couldn't see where the developmental assembly line actually went wrong. Now we have the tools to rewind the tape and find the exact fraction of a second where the biological factory made a critical error.
The diagnostic potential is just paradigm shifting. It moves us from observing symptoms to understanding root mechanical causes. And this raises a highly provocative question, something for you to ponder long after we wrap up today.
Okay, let's hear it.
If we can use 3D to track the exact 4D molecular choreography of a healthy organ assembling itself, and we can clearly spot the early off-tempo signals of a genetic disease causing it to build incorrectly, what happens if we point this 4D time machine at a tumor?
Oh, wow. Because a tumor isn't just a random ball of cells, it is essentially an organ building itself the wrong way.
Exactly. Tumors are highly organized structures. They possess their own cancer stem cells, they build dense structural scaffolding, they actively recruit blood vessels to feed themselves, and they even hijack immune cells.
Right.
They undergo a corrupted, malevolent form of organogenesis. If we could map a tumor's 4D spatial timeline from its inception, could we find the exact right moment in its development, the exact pseudo-time stage where its structural coordination is most vulnerable?
Could we find the specific point in its corrupted choreography to introduce a drug and just dismantle it completely from the inside out?
We could target the exact moment the tumor's orchestra is most out of sync.
That is an incredibly powerful thought to end on. We started this conversation looking at a single, flat 2D photograph of a prequel. And now, thanks to spatial pseudotiming and polyacrylamide hydrogels, we are watching the full 4D movie of life building itself. cell by cell.
It's an exciting time in science, that's for sure.
It allows us to find the exact frame where things go right and the exact frame where things go wrong. Keep asking those deep questions, keep looking for the hidden dimensions in your own work, and thank you for joining the deep dive today.